Hello everyone, my name is Italy Paisley and today we're going to be learning about the protein ATP synthase. But before we get into ATP synthase, I want to give a little background information or a refresher on what exactly is a protein. What is a protein? Proteins are large biomolecules and macromolecules that comprise one or more long chains of amino acid residues. In other words, this means it is a collection of amino acids that do all sorts of things in our body, such as they fight off infections in our bodies, they transport oxygen, and repair and build our muscles. As you can see over here on the left, the top picture is the structure of what protein looks like, and a chart at the bottom that shows the types and functions that protein do in our body. This one shows the repair and build of our muscles, as I mentioned over here. It has insulin hormonal, immunoglobulin, which are antibodies that is used for protection in our bodies, and it does so many other things in our bodies. Proteins is an essential thing that we need in our body. It's our basic building block. So now that you know a little information on what a protein is, we can go into what ATP synthase is and what its structure is. So, ATP synthase is a protein structure that catalyzes the formation of the energy storage molecule adenosine triphosphate, which is ATP, using adenosine diphosphate, ADP, and inorganic phosphate. Now, we want to know, where is ATP synthase located? Where? It is located in the mitochondria. And remember, the mitochondria is a classical role, which is oxidative phosphorylation, which generates ATP by utilizing the energy released during the oxidation of the food that we eat. As you can see over here on the right is a picture of a mitochondria. And then you can see that ATP synthase is right here located in the mitochondria. Because it utilizes the ATP to release energy during the oxidation of the food that we intake. So now we want to take a look at the structure of ATP synthase. And to know about ATP synthase structure is that ATP synthase has alpha helices and beta helices as its secondary structures. And how do you know that it has alpha and beta helices? Well, you want to look at the structure type, and you see you have coils. These coils indicate that it's alpha and beta helices in the structure, which secondary structure consists of beta and alpha helices. So now we want to look at how many peptide chains ATP synthase has. And for us to look at how many peptide chains ATP synthase has, we want to look at how many colors is present. So there's 1, 2, 3, 4 colors that are present. So since there's 4 colors, we have 4 peptide chains that are present, which makes it have a quantitative structure. And quantitative structure is a structure that has 2 or more peptide chains present, which ATP synthase has 4 peptide chains, which is 2 or more of what is needed for a quantitative structure, which makes ATP synthase a quantitative structure. So now that we know about ATP synthase structure and where it's located and how many chains it has, how many peptide chains it has, now we can look at the amino acid sequence of ATP synthase and how we can transcribe it and translate it. So, before we get into the amino acid sequence of ATP synthase, what are amino acids? Amino acids are the building blocks of proteins, and they are extremely important in our bodies. When proteins are digested and broken down in our bodies, they leave behind amino acids. You see, amino acid and proteins linked together. They are the basic building blocks in our bodies. We need proteins and amino acids. They are very, very essential into our lives. So since we know a little bit about amino acids, we can learn about what is transcription, and what is translation before we can get into how we can translate it and transcribe it. So, transcription and translation are the two processes that convert a sequence of nucleotides from DNA into a sequence of amino acid to build the desired protein. These two processes are essential for life. So, now that we know what transcription and translation is, we're going to learn how we can translate the first 10 amino acids of ATP synthase. So we want to look at ATP synthase amino sequence, but we're only looking at the first 10. And we want to translate the first 10, which is right here, into an RNA code. We want to translate it into an RNA code. So, to translate into an RNA code, we want to look at this table, 1 and 3 letter code for amino acid. So, to translate this into an RNA code, we want to look at the first letter, which is F, and match it to what the 3-letter code for the amino acid would be on the table. So F is here, so it will be Fe, and I have Fe right here. Now we want to do the next letter, which is Q, and Q is right here, which is Glen, and we have Glen right here. And we want to keep doing that until we finish it all the way until we have all 10 completed. Now we want to look at the RNA code and how it was translated. So we want to look at this table for the RNA code. So since I have Fe here, we want to look on this table, where is Fe located at, and it's located right here. So we want to use the first one, which is UU, which I have right here. And then we go on to Glen. We want to find Glen on the table, which is right here. And we want to use CAA, which I have here. And you want to keep continuing to do that until all 10 is translated into the RNA code. Now, since we know how to do the translation of into an RNA code, we're going to learn how to transcribe that RNA code into a DNA code. So, one of the rules to remember when transcribing RNA codes into DNA is that DNA has ATCG, RNA has AUGC. So what that means is you want to always pair A to T or U, and then always pair C to G. So to translate the RNA code and DNA code, we want to use, instead of T, we want to use the U. And then we're still going to use the C to G. Because over here, as I've written down, RNA has AUGC, but in DNA, it has ATCG and not U. So the way we look at it is, our first letter here is U. So we want to know what pairs with U, which is A. And I have it right here transcribed into A. Then we go to the next one, U again. We have A again. It's U again. We have A again. Now this one we have C. We're going to pair it with G. And this one we have A. We're going to pair it to U. So you keep doing that all the way until you complete your DNA code. So this is how you would do the amino acid sequence for ATP synthase.